I hope that he's going to do that in his seminar today. But a lot of his colleagues spoke very highly of him, both when it comes to the research that he has done over his career as an instructor, and then just as a great collaborator. And here's just some examples from the packets that we have received in the awards committee. And before I give the floor to Dr. Christensen, I just wanted to thank the awards committee for looking over the nomination packets and just helping out this year. Our administrative assistant, Anna Buffis, who took care of all the plaques and then taking pictures, and for all of you for being here today. We're still virtual, and it means a lot for us that you show up and take time out of your day right before the Eagles game to show up and then enjoy the talk. Thank you. At this point, I will stop sharing the screen, and Dr. Christensen, you can take it away.
All right, well, thank you very much. Let's see if I can do a screen share here.
To verify, are you okay with us recording this talk?
Oh, absolutely. Yes, that's fine.
Okay, awesome. Thank you very much.
All right. Well, yeah, thank you very much. I am indeed honored to receive the Philadelphia ACS Section Award this year. And really, this is not a recognition of me, but it's a recognition of my students and postdoctoral fellows. It really takes a team to advance the science. So I'm honored to receive this award on behalf of the team, both the past and present, the folks in the lab. It's especially meaningful, and especially this year, as I began putting together this talk, I realized that although I arrived at Penn in 1988, as you had noted in the bio, this is actually 40 years of my association with the Philadelphia ACS section. So going back to 1981, I really had my start. The first time I worked in a research laboratory was at Bryn Mawr College. I worked in Professor Charles Swindell's lab, working on a project synthesizing an enzyme inhibitor. I didn't know too much about enzymes at the time, but I learned a lot that summer about enzymes. Also, a project ongoing in the Swindell laboratory was projects toward the synthesis of Taxol, paclitaxel. At the time, a newly discovered potential drug for cancer chemotherapy. So it was really a wonderful time for me during those formative years. I was fresh out of my sophomore year of college. I saw this advertisement for an NSF-sponsored research position at Bryn Mawr. There were three of them, as I recall. It was Charles and Jerry Richmond and Neil Abraham had acquired funding to bring in undergraduates for research. So I got my first experience running organic reactions in, as I recall, sweltering hot laboratories. There was no air conditioning at the time. And I learned that these summers in the Philadelphia area can sometimes get very hot. But while reactions were running, I would sometimes go upstairs and across the way to the library, where I came across Jack Dunnett's book, X-ray Analysis and the Structure of Organic Molecules. And I just became quite fascinated with X-ray crystallography. And learned a little bit more about what Dunnett's was doing with chemical crystallography, learning about organic, the reactivity of organic molecules. And I thought, geez, you can use crystallography to understand chemical reactions catalyzed in enzyme active sites. And so really all of the seeds of what became my career with enzyme structure and mechanism, terpene biosynthesis, and X-ray crystallography, those seeds were planted at Bryn Mawr College in that summer of 1981. So this is 40 years, and a special thank you to Charles Swindell, who I know is logged on this evening. really looking back over these 40 years and seeing how pivotal that summer at Bryn Mawr was for me. I'm very appreciative to you and to the warm welcome I received from all at Bryn Mawr. Was that summer was my first official affiliation with the American Chemical Society, because on the one of the bulletin boards at Bryn Mawr, there was an advertisement for undergraduates to become affiliates of the ACS. You couldn't become a member, as I recall, until you were a graduate student, but you become an undergraduate affiliate. And Professor Ernst Berliner signed my nomination papers to become an affiliate of the ACS. So here, the Philadelphia section, this was my first connection to the American Chemical Society as an undergraduate affiliate some, well, 40 years ago this past summer. So I'm very grateful for that start and for the seeds that were planted that summer in Bryn Mawr. And little did I know I would return a few years later to Philadelphia, where those seeds, they grew into my career, studying enzymes, mainly using X-ray crystallography. We'll talk a little bit, though, about cryo-EM tonight. We study metalloenzymes in one half of my research program, and in the other half of the program, we study enzymes of terpene biosynthesis. So terpenes, it's really the most chemically diverse family of natural products, with more than 90,000 members identified to date in all forms of life. They're quite complex molecules, especially the cyclic terpenoids. And to get to the cyclic terpenoids, one has to proceed along the pathway of primary metabolism, assembling 5-carbon precursors to make isoprenoid diphosphates. They're 10, 15, 20, et cetera, carbons long. But the real molecular diversity arises from secondary, or sometimes called specialized metabolism, that takes these linear isoprenoid diphosphates and it generates a polycyclic product, a hydrocarbon product, oftentimes, with multiple rings and stereocenters. And this happens in a single enzyme catalyzed reaction. And it's remarkable in terms of the carbon chemistry. That is, more than half of the substrate carbon atoms undergo changes in bonding and hybridization during a typical cyclization cascade. While terpenide cyclases are not very fast. In fact, a collaborator, David Kane, once remarked, they make catalytic antibodies look good in terms of their efficiency as catalysts. They nonetheless catalyze reactions that just simply would not occur. It would not occur in the absence of a catalyst. You can't cook geranyl diphosphate under any conditions that will spontaneously yield taxidiene. You need an enzyme to do that chemistry, and this is what I've devoted the past 25 years of my life to, trying to understand the role of the enzyme in that chemistry. So to understand the cyclases of secondary metabolism, we first have to understand the prenyltransferases of primary metabolism. And a lot of this work had been set forth by Popiat, Cornforth, Poulter, and Rilling decades prior to my entry into the field. It's an intermolecular condensation reaction, dimethyl allyl diphosphate and isopentenyl diphosphate. These are the C5 precursors. The ionization of DMAPP is triggered, yielding an allylic cation that then forms a bond with the IPP molecule, resulting in a tertiary carbocation that undergoes an elimination to yield the chain elongated product. Now, this is a process of enzyme. This cycle will keep on going as deep as the pocket will allow. So you do another round of catalysis, you'll generate farnesyl diphosphate. In farnesyl diphosphate synthase, the pocket is this deep. You can't add any more 5-carbon isoprene units on there because the pocket won't allow you to. So really, the enzyme is a one-dimensional template or a ruler. It dictates how many 5-carbon isoprenoids that you can add to that growing chain of the isoprenoid diphosphate. The mechanistic details have been worked out by the scientists listed below. Some key aspects of this that metal ions, magnesium ions usually, but sometimes manganese, occasionally cobalt, are required for the chemistry of ionization to pop the diphosphate group off. And the elimination of the proton is stereospecific. Hans Rilling mused many years ago that it was the inorganic pyrophosphate co-product that does that job, serves as the general base. And structural studies indicate that he was right, with a tremendous foresight. The substrate itself, the product, the co-product, serves a role in catalysis. The crystal structure of farnesyl diphosphate synthase was solved by Jim Saccatini with Dale Poulter in '94. And you see a dimeric architecture here, a dimeric quaternary structure. There's two enzymes. It's oriented like my cup of coffee. The entry to the active site is from the top here into one active site, from the top here into the other active site. Structural features of FPP synthase of note is a characteristic capping helices that cap the active site. These sort of almost at a right angle, poised to cap the active site. So I put little Red Sox caps in honor of my favorite baseball team to try to drive home that point that it's important to cap the active site when you have this reaction running in there, because you're dealing with reactive carbocations, the allylic cations, the tertiary carbocation. If they're exposed to bulk water, you'll quench the reaction. So you need to protect those carbocations from aqueous solvent to avoid that quenching. Now, in terms of the metal ion requirement for catalysis, folks had noticed that many years ago that there were characteristic metal binding motifs, aspartate-rich motifs. They're called DDXXD. These are found on helices D and H, and these bind 3 magnesium ions. So we can look at structures of a variety of prenyl transferases that catalyze these chain elongation reactions. And we see it comes in 3s. So 3 metal ions are coordinated by the diphosphate group of the substrate, or in some cases, the phosphonate group of an inhibitor, or inorganic pyrophosphate itself. And there's 3 metal ions. There's also 3 basic residues, positively charged lysines and or arginine side chains that are required to provide that electrophilic pull to pull that diphosphate leaving group off of the isoproenide diphosphate to generate that initial allylic cation. So now that we know how to make these, these long isoprenoids, the isoprenoid diphosphates through a primary metabolism, this is where my interests perk up here, because I asked the question, how do you go from these linear achiral molecules in one enzyme catalyzed reaction? You generate a hydrocarbon product with multiple rings and stereocenters. So how does that happen? We've solved many structures over the years of enzymes that cyclize 10, 15, and 20 carbon precursors. We've also studied cyclases that don't know how to count. Methyl isoborneol synthase works on a methylated geranyl diphosphate at 10 carbons plus 1 equals 11. Geosmin synthase catalyzes a fragmentation reaction. You start out with a farnesyl diphosphate with 15 carbons that it undergoes a cyclic intermediate, undergoes a transannulation and fragmentation to yield geosmin and acetone. We've also studied, I suppose, an honorary cyclase. It's really not a cyclase, it just catalyzes an elimination reaction from dimethyl allodiphosphate, and that's isoprene synthase. So each of these enzymes, the structures tell a story that has advanced our understanding of terpene cyclase structure and function. And the story began with the cyclases, as one of the quotes provided in the introduction, with pentylenine synthase, structure that was solved in '97 by former student Charles Lesberg, now at Merck. It's a bacterial enzyme, so a single domain enzyme. It too had an aspartate-rich motif, the DDXXD motif, but on helix H, it had a variation or a divergent evolution away from that motif. There's aspartates in it, but it had evolved to residues that are employed for metal coordination, either asparagine or aspartate, serine or threonine, and glutamate for metal coordination. Another thing we noticed when we solved this structure is the resemblance between pentylene synthase and farnesyl diphosphate synthase. So these enzymes have an identical topology, although it may be hard to see from this view. The identical topology, despite insignificant amino acid sequence identity, indicates that these two enzymes, one that makes FPP, the other that cyclizes FPP, these enzymes evolved from a common ancestor early in the evolution of terpene biosynthesis. So this was quite fascinating just to understand the structural biology and the evolutionary biology of terpene biosynthesis as evidenced by these three-dimensional crystal structures. Now, the role of this alternative metal binding motif in the cyclases is the same, whether it's two aspartate rich motifs or one aspartate rich motif and one what we call the NSE motif. The role is the same. You bind three metal ions and you have three basic residues, arginines and or lysine residues, that interact with the diphosphate group, either of the product, the inorganic pyrophosphate, or the diphosphate group of the substrate. And the three metal ions, the three hydrogen bonding residues, provide the electrostatic pull to initiate ionization of the substrate to generate the allylic cation that then reacts with one of the remaining pi bonds in the substrate. So it's really just like the prenyltransferase reaction, only it's an intramolecular condensation reaction, not an intermolecular reaction. So what can we do once we ionize farnesyl diphosphate with the three metal ions and the three basic residues? Well, here's where the beauty, really the combinatorial chemistry of terpene biosynthesis comes into play. Because if we assume that a bond, the initial carbon-carbon bond would be made with C1, with the two remaining pi bonds in the substrate, there are four possibilities for the initial carbon-carbon bond forming reaction. And luckily, the enzymes haven't heard of Markovnikov's rule. It doesn't matter if you're not forming the most stable carbocation, as is the case for pentylene synthase, because the enzyme active site has aromatic residues that are oriented just so in order to preferentially stabilize carbocations, whether they're secondary or tertiary, as needed to enable cyclization cascade to proceed. So there's aromatic residues in the pentylene synthase active site that help to stabilize that otherwise less stable secondary carbocation. Now, we haven't done much work with pentylene synthase in terms of protein engineering. Instead, we focus more on epiisocysteine synthase, another bacterial enzyme that arises from an initial 1,6 cyclization reaction. And immediately the warning bells go off if you teach sophomore organic chemistry, because, well, we're starting out with a trans carbon-carbon double bond here, and we don't know trans carbon-carbon double bond in a 6-membered ring. It has to go cis. So there's a couple of mechanistic steps that one has to proceed through in order to achieve that. So there's an ionization, the metal-triggered ionization of the substrate phosphate group. It recombines at C3. That leaves you a carbon-carbon single bond at C2 to C3, allows you to rotate. Once rotated, you reionize, and that narrow-ideal cation, then it can serve as the point of contact for C1, C6 bond formation to generate a bisabollyl carbocation. Now, to make epiisocysteine, this will proceed through additional bond-making reactions, and there's hydride shifts and some carbon migrations in this mechanism that ultimately yield epiisocysteine. Now, how does the enzyme do all this? Again, the enzyme really has to serve as a template first and foremost, because the substrate, the farnesyl diphosphate, any linear isoprenoid diphosphate is going to be very flexible. And per 5-carbon isoprenoid unit, there's 3 carbon-carbon single bonds. And I like to think of an isoprenoid as 50% more flexible than a peptide, right? Per amino acid, you have 2 bond rotations around the phi and the psi angles. Well, an isoprenoid, you've got 3 bond rotations that have to be fixed in place in order to orient, in this case, the substrate properly for the correct sequence of carbon-carbon bond forming steps. So the enzyme active site serves a critical role as a template. And that means the 3-dimensional contour of the enzyme active site is defined by the amino acid residues lining the active site of the enzyme in the middle here. They're going to play a role in directing catalysis, and not necessarily by doing anything during the reaction, other than providing that hardened crucible that will not react with the carbocation intermediates, but hold them and stabilize them in order to allow the organic chemistry of the cyclization reactions to proceed. So Julie Aaron Himmelberger in my lab solved the first structure of epi-isocysteine synthase in the lab. And we also have structures with ligands bound. And it's in this enclosed conformation that we really gain all of the mechanistic information about what the enzyme is doing. There's 3 metal ions that it binds along with a inorganic pyrophosphate. This helps to close the hatch. Now, we don't have the caps like we did in the prenyl transferases, but we have these active site loops that interact with residues that are coordinating to the metal ions, forming hydrogen bonds with those residues and so forth, just sealing off the inside of the active site here occupied by a quaternary ammonium benzyl triethylammonium cation. Now, what does that enclosed active site volume look like? Well, it's very much like the 3-dimensional shape of epi-isocysteine. So in terms of its function as a contour, a template for catalysis, the active site in that closed conformation where carbocations are protected from bulk solvent, that active site serves as a template to enforce the correct folding of the substrate FPP so that it is lined up, all of the proper trajectories of carbon-carbon bond forming steps are lined up as guided by, as enforced by that 3-dimensional template. Now, if this is a template-directed enzyme, then if we change the shape of the template, maybe we can change the chemistry that goes on in that active site. As long as we preserve the metal binding residues, we'll preserve catalytic activity for the enzyme. We won't perturb activity as long as you can bind metal ions, 3 metal ions, the 3 hydrogen bonding groups for the diphosphate group. You can trigger the ionization of the farnesyl diphosphate, but maybe you can force it to bind with a different orientation and turn off epi-isocysteine formation and turn on the generation of an alternative sesquiterpene. And we did just this a couple of years ago. One of the active site aromatic residues, so we can substitute with serine, methionine, glutamine, and we turned off epi-isocysteine biosynthesis and turned on a pathway leading to sesquisabenine with this 310 this strained linkage that you see here. So how did we do that? I mean, what stopped the reaction, what ordinarily would have been the reaction of the remaining pi bond here with the tertiary cation here that would have led to epi-isocysteine formation? Well, if we look at the 3-dimensional contour of the phenylalanine 96 to serine substitution, we see we've opened up volume in the enzyme active site. So by putting a smaller amino acid in this position, there's extra volume that pendant chain, that pendant isoprenoid group can bend away from the core of the molecule where the carbocation would have been in that homobisabulyl cation. So if you're not holding reactive groups close together, you're not going to form that particular carbon-carbon bond. Then moreover, the constriction of the active site where the homobisabulyl cation is nested, that facilitates the transannulation reaction leading to sesquisabinine formation. So Patrick Blank in my lab, a graduate student, asked the question, well, how does this compare with an authentic sesquisabinine synthase? And he prepared and crystallized the sesquisabinine synthase 1 from Indian sandalwood, then looked at the three-dimensional contour of the enzyme, and then did some molecular modeling. So the chicken wire here is just the three-dimensional contour of the enzyme active site. So Patrick showed that the homobisabulyl cation could be modeled such that the six-membered ring is close to where it started from. Here's the diphosphate group in the active site, but here that pendant isoprenoid tail, it's forced away from that homobisabulyl cation, so it cannot react. The pi bond cannot approach. It's constricted. It cannot approach that cation. So the lesson we learned in comparing the epi-isocysteine variant with the authentic sesquisabinine synthase is that it doesn't matter if the isoprenoid tail is constrained to go straight out away from the six-membered ring, or if it's bent. The key point is that any conformation that forces that isoprenoid tail away from the tertiary carbocation will lead to the alternative product to sesquisabinine. So we've really learned the most in our studies of terpene cyclase structure and function from our studies of the sesquiterpene synthases with structures of bound analogs, analysis of active site contours and so forth. But we've also invested a lot of time in the diterpene synthases. And again, hearkening back to the seeds of my education in the Swindell group at Bryn Mawr College, I always had this fascination with Taxol and Taxol biosynthesis. So taxodiene synthase was a diterpene synthase that had always been near and dear to my heart. So we worked on structures of both taxodiene synthase and N-copylyl diphosphate synthase about 10 years ago. And these enzymes provide some interesting contrasts. Even as you look at them from afar, they adopt similar three-dimensional folds, but they make very, very different products. So let's talk about taxodiene synthase first. This taxodiene synthase functions just like the other cyclases we've talked about up until now. There's a metal-triggered ionization of the substrate diphosphate group. Now the substrate is geranyl diphosphate, GGPP, a 20-carbon isoprenoid. Again, 3 metal ions are required for activity. The ionization of the substrate diphosphate group triggers a cascade of carbon-carbon bond-forming reactions. There's an intramolecular proton transfer, transannulation reaction, ultimately leading to the taxine phoryl cation that is quenched by a general base that is very likely to be inorganic pyrophosphate, as Hans Rilling speculated so many years ago about the prenyltransferases, the catalytic importance of pyrophosphate as a general base in the enzyme mechanism. So let's give these types of enzymes a name now. When you trigger catalysis using the trinuclear metal cluster, we're going to call this a class 1 terpenoid cyclase. So pentalenine synthase, epi-isocysteine synthase, taxodiene synthase. These are class 1 terpenoid cyclases. How does a class 1 cyclase work? Well, just like the others. 3 metal ions bind in the enzyme active site. There's the metal binding motifs, the DDXXD and NSE motifs in that class 1 active site. The binding of the metal ions in the diphosphate group of the substrate analog here triggers conformational changes that enclose the active site. Taxodiene synthase is a little bit different though, because we have these other domains kind of hanging on. And what do they do, you might ask? They do absolutely nothing in terms of the chemistry of catalysis. They're an evolutionary vestige, the beta domain and the gamma domain, which resulted from a gene duplication and fusion of the beta domain. This is actually inserted between the first alpha helix and the second alpha helix in the beta domain. So we have a vestigial domain, but why is it there? Well, it's an evolutionary remnant for taxodiene synthase. Now let's talk about N-copolyl diphosphate synthase. This is a class 2 terpene cyclase. And it's a class 2 cyclase because it doesn't use metal ions to initiate initial carbocation formation. And there's a magnesium ion that's implicated for diphosphate binding, but there's no chemistry that occurs on that end of GGPP. Instead, a class 2 cyclase initiates catalysis by protonating a carbon-carbon double bond, typically at the other end of GGPP, and that initiates a carbon-carbon bond-forming cascade that yields to the final product with that diphosphate group intact on the product. Well, the three-dimensional structure of N-copolyl diphosphate synthase reveals that same alpha, beta, gamma domain architecture. But here we see we don't have the metal binding motifs. There's no red and orange here in the blue domain. So here that class 1 domain is an evolutionary vestige. The class 2 active site, however, is active because here there's an aspartic acid general acid marked by the sequence DXDD. So this is not a metal binding aspartate now. This is a general acid aspartate. The middle aspartate here is the general acid that serves that protonation function in protonating the terminal carbon-carbon double bond of GGPP. And here you see a bound analog marking the position of the active site in that class 2 cyclase domain. So really, when we compare N-copolyl diphosphate synthase and taxodiene synthase, this is the first step, really the first step in making remarkably different products, remarkably diverse products, starting with a common substrate. It begins with that first step in how you initiate the cyclization reaction. If you want to initiate your initial carbocation on the isoprenoid end of GGPP, you use the class 2 active site, and then that initially formed carbocation can react in sequence or in concert with the remaining pi bonds of the substrate. Or if you want to initiate catalysis at the diphosphate end of the substrate, you use the class 1 active site with its 3 metal ions to pop the diphosphate group off. And then that allylic carbocation can react with the remaining pi bonds of the substrate in the cascade of reactions that ensue. So this is the first step in molecular diversity in terpene biosynthesis. So our interest in diterpene synthase continues even to the present day. So now some 10 years after our studies of taxodiene synthase and N-copolyl diphosphate synthase, we've really turned our attention to assembly line diterpene synthases, which are oligomeric nanoassemblies that combine the prenyltransferase and cyclase activities. So remember, the prenyltransferase is what assembles the 5-carbon building blocks to make GGPP, and the cyclase cyclizes GGPP to make different hydrocarbon products. So systems that we have studied recently are copolyl diphosphate synthase. So this is a different enzyme from the N-copolyl diphosphate synthase I just showed you. This makes a different stereoisomer of copolyl diphosphate. And we've also studied fucicosadiene synthase, which we've now seen in 2 quaternary structures. Excuse me, hexameric structure here, and the large and dynamic octameric structure shown down below. So let me introduce you to these enzymes. I'll start by talking about copolyl diphosphate synthase. This enzyme was discovered by Ikura Abe and collaborators. And here it's a bifunctional enzyme. We've got the prenyltransferase in the alpha domain and a class 2 cyclase at the interface of the beta and gamma domain. So just like the structure of N-copolyl diphosphate synthase that I showed you previously. But the big difference here is how these domains are linked. Sure, the domain architecture is alpha, beta, gamma, as you see here. But unlike N-copolyl diphosphate synthase or taxodiene synthase or other enzymes that have subsequently been studied, instead of having a 33 to 40 residue linker connecting the alpha and beta gamma domains, in copolyl diphosphate synthase from Penicillium verruculosum, we have a 170 residue linker. So 4 or 5 times longer than the normal linker that would connect an alpha and a beta gamma domain. So, wow, is it ordered? Is it not ordered? We don't know. And this is how I attracted postdoctoral fellow Trey Ronnebaum to the lab, to come in to the lab and study Penicillium verruculosum copolyl-diphosphate synthase. How does it work? What is its three-dimensional structure? Well, it turned out we couldn't crystallize the full-length protein, but Trey was able to crystallize and determine the structure of the prenyltransferase. The characteristic structure of the prenyltransferase, dimeric architecture with our little alpha helix caps over the active sites. So we see basically the hexameric architecture, quaternary structure of the enzyme. It can be described as a trimer of dimers, where the fundamental quaternary structure unit is this dimer. And just like the dimer that we saw for farnesyl diphosphate synthase back in 1994, that same quaternary structure, but the GGP synthases are special. Those dimers can tend to oligomerize, as was first observed for human GGP synthase. Again, dimeric core architecture, but 3 of those dimers assemble to form a hexamer. So, unable to crystallize the full-length enzyme, Trey struck up a collaboration with Kishore Gupta at Penn School of Medicine to determine the molecular envelope of the full-length enzyme using small angle X-ray scattering. And that molecular envelope is what you see here, outlined, this little cloud that's outlined. And in modeling SACS data like this, well, Trey took that hexamer of the GGPP synthase, the prenyltransferase of copolyl diphosphate synthase, put that in the center, and then modeled very easily the beta gamma domains in this starburst-like architecture, the quaternary structure of the full-length enzyme. And remembering too, we have linkers connecting the alpha domain with the beta gamma domains here, and those linkers are largely disordered or conformationally flexible, and that's evident in the experimental data. A very flexible system. So a very flexible system and a very large system, because the diameter of copolyl diphosphate synthase, the diameter of this hexamer, is about 300 angstroms. And this evokes all sorts of questions about, well, is it an assembly line cyclase? The substrates go in, the 5-carbon substrates go in here. We have a central factory, a GGPP synthase factory that makes GGPP. The GGPP then transits out to the cyclase domains for that class 2 cyclization reaction. How does it do that? Well, we don't know all of those details yet, but we are able to ask a lot of very interesting questions about substrate transit and even the role of this globule-like intrinsically disordered region, the linker that connects the alpha and the beta domains in this assembly. Now, running this through structure prediction algorithms, one can see little bits of alpha helix in there, but it's mostly, it comes up as random coil and a disordered random coil. Yet it is also predicted to be a globule-like, not an extended alpha helix or an extended beta strand-like conformation, but a globule, a very dynamically behaved globule of protein connecting the prenyltransferase and the cyclase. And what we have found very interesting and work done by Trey and Sam Eaton and Emily Bracken in the lab is that you can mutate that catalytic general acid in the beta domain, and that effect is felt in the prenyltransferase domain with a smaller kcat. So we can compromise catalysis a bit. I mean, tenfold's not a lot, but it's measurable and it's real. How do these domains communicate with each other through this largely disordered molten globule, like a bit of peptide in the middle? I don't have an answer to that question, but perhaps the future graduate students in my group will be answering that question in the years to come. So This gives us a different perspective on terpene biosynthesis, getting beyond the individual cyclase enzyme, but now thinking about the enzyme and the pathway as the enzymes can interact with each other. And here they have to interact with each other because they are, after all, covalently linked in the same polypeptide chain. Now, a copolyl diphosphate synthase is an example of an oligomeric alpha beta gamma enzyme. In contrast, fusicosidine synthase, it's an enzyme in which two alpha domains are connected to each other by a flexible linker. And again, we're dealing with a prenyltransferase that generates GGPP. It's a C-terminal prenyltransferase. And then GGPP transits to the cyclase domain, where it undergoes cyclization to generate fusicosidine. And these are the first committed steps in the biosynthesis of fusicosin A, an emerging lead for cancer chemotherapy and nerve regeneration, or a couple of potential pharmaceutical applications. Mengbin Chen, a graduate student in my lab, who graduated in 2016, she was the first to attempt to crystallize the full-length fusicosidine synthase. We couldn't get crystals of the full-length enzyme. But what Mengbin did was prepared constructs of the individual domains, the prenyltransferase domain. She crystallized it and saw what is now a very familiar structure to us, this hexamer, or the trimer of dimers. It's the prenyltransferase. You see the characteristic capping helices over the top. Mengbin also solved separately the structure of the cyclase domain with a bisphosphonate inhibitor bound, with its 3 metal ions. Those are coordinated by residues from the aspartate rich and the NSE motif. So we learned a lot from these studies, if not about the assembly of the full-length protein, certainly about structure mechanism relationships in the enzyme active sites. So for example, you're remembering the template function of the cyclase. Mengbin showed that the 3-dimensional contour of the active site was very product-like. So it makes sense that the cyclase has a product-like contour that helps to chaperone the folding of GGPP in a product-like conformation prior to initiation of that first carbocation intermediate to initiate the cyclization cascade. Now, fusicosidine synthase, it's a somewhat promiscuous enzyme. Fusicosidine is the major product. It makes 2 other products. One of them has not been characterized. The other product is generated to a small degree. And this cyclization promiscuity is observed both in full-length PAFS as well as just the catalytic domain, the construct we term PAFS 350. The structure-function relationships for both are the same, whether it's connected to the full-length protein or not. So, unable to crystallize the full-length enzyme, Mengbin resorted to small-angle X-ray scattering to determine a molecular envelope for the full-length protein. And she got a really good fit to the molecular envelope. This is very low resolution, but she was able to model in the central prenyltransferase hexamer with the associated cyclase domains on the side, in order to account for the fit to the observed data for the small angle X-ray scattering. So here it looks, based on this model, we have something we can test. We can do enzymological studies to help illuminate the function of this bifunctional assembly line enzyme. So based on this model, Golda Harris in my group, she asked the question, these enzyme active sites, well, the enzymes seem to be very close to each other. Is there channeling? And if you've got a bifunctional enzyme and things are very close together, it's a very logical question to ask. And Golda designed an experiment that utilized substrate GGP so as to compete with 2 different diterpene cyclases, the fusicosidine synthase and taxodiene synthase. So if you take an equimolar mixture of the 2 diterpene synthases, treat them, incubate them with GGPP, you'll generate both hydrocarbons, fusicosidine and taxidine, with a 4:1 ratio. You get a little bit more fusicosidine because the catalytic efficiency, kcat over km, that's a little bit bigger for fusicosidine synthase compared with taxidine synthase. Now, the second experiment that Golda did was take the 5-carbon precursors, DMAPP and IPP, and incubated the reaction mixture, the enzyme mixture, just with these 5-carbon precursors, so that the only source of GGPP would be that generated by the prenyltransferase of fusicosidine synthase. Now that GGPP, if it's competing with both enzymes equally, we should get the same product ratio of 4:1, fusicosidine, taxidine. But that's not what we saw. We saw a product ratio that was 46:1. And that told us that most of the GGPP that's generated on fusicosidine synthase stays on fusicosidine synthase. So wow, now the plot thickens. We need a higher resolution structure than this 25 angstrom resolution molecular envelope from small angle X-ray scattering. So what can we do? This is when Jackie Falo took over the project, and Jackie said, well, we have to do a cryo-EM structure of fusicosidine synthase. Jackie did negative stain electron microscopy, followed up by cryo-electron microscopy. We saw some things that looked familiar. We saw some things that didn't look familiar. So in the negative stain EM, we saw things that looked like tetramers. I saw these tetramer looking things in the cryo-EM as well. We also saw trimer looking things in the cryo-EM as well, but we saw about 90% tetramer and at about 10% trimer. Well, let's call this an apparent trimer, an apparent tetramer. Because there were more particles that looked tetrameric, Jackie worked with these to make a 3D reconstruction. And got a beautiful 3.99 angstrom resolution reconstruction of these apparent tetramers, as you see here. And it's nice alpha helices. You see the bands, if you will, of revealing the pitch of the alpha helices here. Beautiful structure. But gosh, if you look at this structure, this bit here and this bit here, this bit here, this bit here, those look like the caps of the prenyltransferase. And indeed, those are the capping helices of the prenyltransferase. So what we have is not a tetramer, it's not prenyltransferase, but cyclase, but instead it's an octamer of prenyltransferases. Wow, so it's an octamer of prenyltransferases. Well, that's something you don't see every day. And then we were a little bit despondent because we're missing half of the protein. Where are the cyclases? I spent 25 years studying cyclases. Where are the cyclases? So we were puzzled by this. Oh, maybe the enzyme fell apart. Maybe there was proteolytic degradation. Jackie went back, checked samples. There was no degradation. The cyclase was still there. We did the assay for cyclase activity. We made fusicosidine just fine. So where's the cyclase? And so we went back to the negative stained images of fusicosidine synthase. And you see these little donuts now that we know what we're looking for. These little donuts are the, here, donut there. Those donuts are the prenyltransferase core, mainly the octamers, but some hexamers in there. Take a close look at some of these. And with the negative staining to help highlight the protein surrounding those cores, get a spot here, spot there, spot there, spot there. Here's spot, 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 spot. The cyclase domains are there, but they're splayed out away from that prenyltransferase core. And not only are they splayed out, they're splayed out in different positions, such that when you do the averaging, you're basically, in essence, superimposing all of the particles. The cyclases predominantly average out in the reconstruction, the 2D reconstructions, and then the 3D reconstructions. So we were able to trap the cyclases in more ordered conformations, seeing them at lower resolution by using chemical cross-linking. This is the quality of the image that we see in these reconstructions. So we see positions where a cyclase can cap the central pore of the prenyltransferase, and the cyclases can also adopt positions on the side, on the hip of the prenyltransferase octamer. And before she left the lab for her postdoc at ECST, Jackie showed that you can also achieve these cyclase-associated modes in the absence of chemical cross-linking, but in the presence of a bound inhibitor. And we're wondering if perhaps the cyclase and prenyltransferase adopt more associated modes in the presence of substrate while doing the job of catalysis. So this led us to revise a model for structure and dynamics of fusicosidine synthase, where the cyclase domains can adopt splayed out positions relative to the central prenyltransferase, or they can adopt more closely associated positions. And one can imagine a scenario where GGPP is released into the central cavity of the prenyltransferase and then diffuses by proximity into a capping cyclase domain. Alternatively, GGPP can be extruded from that central cavity and merely diffuse, perhaps electrostatically guided to a cyclase domain if the cyclase domain is associated on the side, or even make its way through cluster channeling, so-called cluster channeling. GGPP exiting that central pore, there will be a high probability that it will encounter at least one of these cyclase domains before it has a chance to diffuse further out into solution. So these are possible models for channeling in the fusicosidine synthase reaction, perhaps a combination of proximity channeling or cluster channeling. I know enzymologists get really hung up over the use of the word channel. When you say channel, you think, oh, a channel where you can drive through, like a tunnel. So maybe a better word to describe this channeling is directed substrate transit, whether it's by virtue of statistics of cluster channeling or by virtue of proximity. It's just something that enables more efficient use of the product GGP that comes out of the prenyltransferase destined for the cyclization reaction. So seeing that it's 5 minutes to 8, and I'm reliably informed that the Eagles game begins at 8:05, let me tie things up. We've covered a lot of ground this evening. The story of terpene cyclase is a story of domain architecture, the alpha and beta and the gamma domains. So the lesson comes in threes. The story of the greater issue of assembly line cyclases is a story of not only domain architecture, but oligomerization, because it's the oligomerization that yields these large nanoscale factories for terpene biosynthesis. And we envisage a day when we can engineer not only the prenyltransferase to make GGPP, or as Trey and collaborators have recently shown, we can convert the GGPP synthase into an FPP synthase. We can hook up these prenyltransferase factories with splayed out cyclases, designer cyclases. We can switch different class 1 enzymes. We can switch different class 2 enzymes on that central chassis of an octameric or a hexameric prenyltransferase factory in the center. Not only that, we don't have to limit ourselves to naturally occurring cyclases for this work, but we can use engineered cyclases to generate cyclic terpenes for which no naturally occurring enzyme has yet been identified. And that, I think, is the true power of protein engineering in biosynthesis of these natural products. So that's the end of my terpene cyclase talk. Let me just thank the folks who did the work that I talked about this evening. I thank the NIH for their support of our terpene biosynthesis program. I'm especially grateful to my colleagues at Penn for these past 33 years, being such wonderful colleagues, as students that I've had at Penn, whose work is being recognized this evening. And especially thankful for my formative years, 40 years ago at Bryn Mawr College, that made Philadelphia seem, and the Philadelphia area seem like home. It seemed like I was destined to return, and I'm glad I did. Thank you very much.
Thank you. That was a wonderful talk. I really appreciated it. At this point, we can open it up for questions if anyone has any. while we get started, I'll actually ask one. So I was really glad that you had all the structures of all the different terpenes on there. I was curious how the naming convention worked, because I couldn't really quite follow how you would name the different terpenes themselves as you kind of made the different transformations.
The cyclic terpenes, I don't know what convention is used by the folks who discover them. There must be some convention. I think the most important thing is not to overlap with other names. Because I remember having this conversation with Dale Poulter once. There's a curcumine synthase, CURCUMENE, and there's a curcumine, right, missing by, it's just different by just one R, one letter. And I said to Dale, geez, yeah, maybe we should change the name. And he said, well, no, just, people know how to spell. Well, so based on that one example, I think it's a matter of, at least with the informal names for the compounds, so not strict EPAC names for the compounds, there doesn't seem to be a rhyme or reason for it. But with regard to the general name of a hemiterpene, a monoterpene, sesquiterpene, diterpene, those correlate with the numbers, the actual number of carbons in the terpene. So at least there is logic in that part of the nomenclature.
Excellent. Thank you very much.
So David, how close are you to inserting an artificial construct to replace one of these cogs in your wheel?
Oh, we can do that. We haven't done it yet with the bifunctional systems, but we have, as I mentioned, we've converted the GGPP synthase into an FPP synthase. So that's which is published in Biochemistry. But it's really trivial to splice off one enzyme and add another. So this is on the docket for future study.
That's really beautiful. Thank you so much.
Thanks.
David, where did you do the cryo work?
At the Pacific Northwest Cryo EM Center. An old college classmate is the director of that center. So he said, come on out. So I sent Jackie out. She went out a couple of times. We've also taken cryo EM data down at the facility at NCI. It's free. They're national facilities.
Right.
In comparison with Penn, to do a full data collection at Penn, it's about $3,000. And I like to be, I like to count pennies when we do our science.
Very nice. Beautiful story.
Thank you.
Yeah, it was really great.
And I'm delighted to see Charles Swindell here as well. 40 years. That was something.
Yep, very nice, David. Very nice.
Thank you.
So I guess I have another question for you is, how nice does your lab smell? I know a lot of terpenes smell really, really pleasant. Is your lab have mostly a pleasant smell from all the terpenes that you're end up making?
Mostly, except when we were doing the work with methyl isoborneol synthase and the geosmin synthase, but really mainly the methyl isoborneol synthase. God, that stuff stinks. And it's the methyl isoborneol is one of the contaminants in the that makes fish smell bad and water give that off flavor. So that was a rough time in the lab, but you do your assays in the hood to minimize that.
Nice. Well, I just pause for a second to see if there are any other questions for David. And if they're not, please everybody join me in thanking David for an amazing talk, and congratulations again. That was wonderful. So
Thank
awesome
you.
job.
Thank you all.
All right, cool. Well, yeah, at least you have your plaque, and hopefully you'll be getting something else in the mail too. So thank you very much for the talk tonight.
It arrived today. Thank you very much for that.
It's great. And thank you, Ivana. I know you've done a tremendous amount of work leading the awards committee. To
Yeah,
get to
no
this point.
problem. We hope that next year this event can be in person. So if it is, we'll invite you to come over and enjoy dinner with us, because that's usually part of this. But unfortunately, this year that did not happen.
Right. Well,
Yeah.
here's hoping that next year will be, we're in a transitional year right now.
Mhm.
Hopefully we'll keep transitioning for the better.
As always, right?
Yeah. I'm giving my first in-person talk at the end of the month at Boston University. So it's slow. The transition that's beginning to happen now. A little bit nervous with the case counts being what they are across the country, but then again, it depends on where you're going and what the circumstances are of the visit and whether they're following appropriate safety precautions or not. So
Are you driving or are you flying?
I'm going to drive. I'm going to drive.
That'll make it a lot safer, right?
Yeah, but I am going to go to an enzyme meeting in January in Arizona, and it's too far to drive. So I will be flying for that. But my son has come back and forth a few times now from California. It's a long flight. He says everyone's wearing masks and so forth, and it seems pretty safe. So we'll all see how it goes.
Well, I wish you good health and a good time during your travels.
Yeah, thank you. Thank you.
Yeah. Excellent. All right. Well, thank you. I'll say good evening now, but thank you very much for excellent talk, and to all of you for joining tonight.
All right, thank you.
Here, here.
Thank you, David.
Thanks,
Thank
David.
you. Thanks.
Thank you.
Bye-bye.